Hello, my name is Alexsis Garcia. I am an undergraduate researcher for the Department of Biological Sciences, and I will be presenting the pseudocooperation of Pseudomonas aeruginosa and Staphylococcus aureus mediated through purine exchange. So to start off, cystic fibrosis is a genetic disorder that is caused by a presence of mutations in the cystic fibrosis transmembrane conductance regulator, or CFTR. And for those of you that don't know, CFTR is actually very important. It causes mucus in the lungs and the lining of the airways to ensure that any particulates that come in through breathing do not make it into the lungs. So if you notice on the reference 1, where we see a normal airway with a thin layer of mucus, which I explained earlier, that it's to make sure that no particulates that are that enter our respiratory system don't make it all the way through to your lungs. And underneath it, we see an airway with cystic fibrosis. As you can see, there's a significant difference between the lining of the mucus as well as the little areas of green where we can see that it has had a bacterial infection. So cystic fibrosis is known to have bacterial infections and in times chronic infections. This is mainly just due to the fact that cystic fibrosis is known to have a very rich in nutrient-rich environment for these bacteria. So in a previous study, we noticed that the CF isolates were not killing staph. And we noticed this when we had inoculated a S. aureus lawn with a CF isolate, which you can see on the right-hand side that there was significant growth. The center is a disc that was uninoculated, and on the left is a disc that was inoculated with a PA14, which is Pseudomonas aeruginosa. And you can see that there was significant lysis of the S. aureus, but when you compare the left to the far right, you can see that there was growth of the S. aureus when it was paired with a CF isolate. So due to this, we believe that P. aeruginosa and S. aureus possibly complement each other within a polymicrobial environment and increase their survival through cooperation. So the main purpose of this study was to see if P. aeruginosa and S. aureus actually complemented each other, and the reason for this was we understand and know that P. aeruginosa is very opportunistic, but for some reason, as we saw in the initial slide, that there was a reduced lysis of S. aureus within the CF isolate. So when we were conducting this experiment, we chose the parent strains of Pseudomonas aeruginosa and the Staphylococcus aureus, and along with those, we used the transpotent mutant strains that were deficient with purine. And for each of these, we cultured them overnight with their selective media, which for the PA14, we use Lysogeny broth, or LB. And for the S. aureus, we use the typic soy broth, or TSB, which were washed 3 times with 1 times PBS, which PBS is the phosphate buffered saline. And afterward, we suspended the cells to OD 600 and later diluted 100 times in a 96 well plate that was predisposed with RPMI media with 1% amino acid. And RPMI media is the Roswell Park Medical Institute. So after we inoculated and diluted and incubated for 48 hours at 37 degrees Celsius, we inoculated each of these cells into the selective agar plates. So for PA14, we used the cetrimide agar plate, and for the staph, we used the metronol salt agar plate. And We also incubated and cultured these in both monoculture and co-cultured with the parent strains and with the purine deficient strains. So what we noticed is that the co-cultures actually exhibited a rescue growth. So when you compare the GE2 mono from figure 1 to its counterparts, the GE2 co-cultured with PA14, which is both of these are the parent strains, you can see that there was a significant amount of lysis when compared to the monoculture, as well as the strain that was grown with the purine deficient. You can also see in comparison to the PA14 monocultures, as well as the PA14 co-cultured with the GE2. And we also see the difference with the PRC deficient grown as a monoculture, as well as the PRC co-cultured with the GE2. And you can see on the figure 2 is the purine deficient of GE2, which is the staph with mono, as well as the staph grown co-culture with the parent strain. And you can see that there was a significant amount, there was a significant difference between the two. And then lastly, you can see the purine deficient strain grown with the purine deficient strain of PA14. So we also wanted to see if this phenomenon was actually due to eDNA released by Pseudomonas aeruginosa mediated lysis or autolysis. So to do this, we used herring eDNA to measure the amounts of eDNA present in the monoculture and co-culture to see if eDNA promoted the rescue. So we repeated the same process as we did with the initial cultures. The only difference is that we added 0, 10, 100, 300, 600, and 900 micrograms per milliliter of herring sperm DNA with DNase enzyme one-time solution for 37 degrees. And as you can see, the concentration of each one. So the GE2 mono showed less concentrated eDNA, whereas the PA14 mono showed a little bit higher concentration, and then the PA14 and GE2 mono had a significantly higher concentration of eDNA, and the PRC transpotent mutant with the GE2 showed less compared to the PA14 and GE2. So from this, we concluded that the rescue growth in the co-culture with the mutant strains of P. aeruginosa and S. aureus, along with their respective parent counterparts, is possible. We also believe that the natural auxotrophies within some metabolic pathways could potentially be indicators of intra and intermetabolic complementation. And these could indicate that there's an evolution of cooperative and synergist interactions within polymicrobial communities. Because of this, we believe that there is potential in targeting other metabolic pathways to confirm the results of this. The data from this can actually justify targeting the bacteria's resources to overcome the antibiotic resistance factor that is shown by polymicrobial communities. And we also believe that intra and interspecies metabolic interactions should continue to be studied to further understand the extent of the pseudo-cooperation. And this requires further experimentation before we can even attempt to overcome the antibiotic resistance factor. I would like to give a giant thanks to the NIH for grant funding, along with the Plains to Bridges Baccalaureate program, along with Dr. Jacqueline Keynes-Garrel and Dr. Lacey Alexander for partnering me up with Dr. Wakeman, as well as the Wakeman Lab. I would like to thank Dr. Wakeman for the mentorship and guidance. And lastly, Hafiz and Jawasmika for mentoring me throughout the project. And Lastly, if any of you have any questions or any feedback, please feel free to contact me, and thank you for watching.